All right, so we are getting close to our first exam. All right, so you guys for this lecture will still have a quiz, just like we've been doing. But following this week, then you're gonna have your first exam. So what that is obviously there's not gonna be any lecture posted. It will be strictly on the assessment page of Blackboard, just for all the quizzes I've been. All right, and then it's going to be the same exact format, all multiple choice, 100 questions. Use your browser, your lockdown browser. All right, and I would give yourself, at least like an hour. Okay, so it is a one-time sit down, so set up some time if you need it. Um, but I would at least set time for about an hour. So on to the next system. We're going to start talking about what's known as the immune system. But to understand the immune system, you know what immunity is. So I'm sure you've all have heard immune system, immunity, things like that. And in the quick summary, it's just that our body, either internally and externally, we're surrounded by these potential harmful things like your viruses, your bacteria, your parasites, all these microorganisms that can cause harmful things to our body. And we have to be able to, you can say, combat them to keep our homeostasis, right? Keep us surviving and thriving. And it really depends heavily on the lymphoid or lymphatic systems. Right? That's what helps to produce our immune responses. Now, strictly the term immunity, that just refers to the body's ability to defend itself. All right, to defend itself against what are known as pathogens or microorganisms that can cause us harm. Now, when you're talking about immunity, it isn't just like immunity is this term that covers everything. There's different kind of subsets of immunity known as an innate or non-specific immunity, adaptive, or also known as a specific immunity. And we'll get to covering both of those a little more further. Now, lymphoid system, it's important in all of them. Okay, it's not just like, all right, this innate immunity uses lymphatic system, but adaptive does. It's actually plays a pretty central role in adaptive immunity, right? So it's pretty key there, and it does support some aspects of the innate immunity. Okay, and we'll cover exactly what ports of that it does. All right, but you also see as we go through is that it works pretty tightly with other systems, such as the cardiovascular system, which makes sense, right? Because if we're having an immune reaction or an immune response, we want whatever response to be going through our system as quick as possible to get to where it needs to be. So it makes sense that it's going to have a tight connection with the cardiovascular side of things. All right, so starting out in innate immunity, okay, we have this as present at birth. All right, so this is more of an immediate. It's got a non-specific defense against all pathogens, so it's not so forth targeting a specific pathogen. It's there to kind of put the frontline soldiers down. Okay, and so because of this, it has some ups and downs. Okay, one is since it's not specific, it can't really have this specific, I guess you could say, response to whatever invaders coming into the body. It's just going to treat it the same way as it would treat everything else. All right, so there's no guarantee that that way is going to help. Now, innate immunity, like I said, that's what we have at birth. So this is why, usually when babies are born, you try to be a little more protective of them. This is why they get their vaccines, because the innate immunity can't really take all of it on. All right. Now, this includes, like I said, it's called the first line of defense, not just cells. All right. And actually, with innate, there's not really a lot of cells that come into play. I guess there's some, but not really a lot. And a couple of the first lines is our physical and chemical barriers. Your skin is considered a part of the innate immunity. Okay, so as well as your oil in your skin, other types of chemical barriers that are associated with membranes. But you also have then what is known as the second line of defense. Now that is when we start to get into cells. All right, so skin, outside, first line defense. The second line of defense is when we start to get the cells of the innate immunity. Okay, so these are internal defenses. So we've now gone inside the body. Now this has a lot. It's got antimicrobial substances, the cells. Probably have heard natural killer cells, and our other associated, things like fever. All right, so fever obviously is a symptom of an inflammatory reaction. It's got its own benefits with helping to fight the virus or whatever is inside. Okay, and innate immunity, like I said, is kind of like the first line defense, kind of the first response, and as so, it acts as a early warning system as well as a rapid response team. Okay, so I like to think of the innate immunity as kind of like the police officers that get on scene, whereas the adaptive immunity is the SWAT team that had to get prepared to show up. All right, so you got one that covers it all, can do everything, but you got one that has a specific response to the other. All right, but our innate immunity is the one that gets there first. Adaptive immunity. Okay, so it develops over time. It's basically very key to what exactly vaccines are and what they do. So it's just a type of immunity that develops to exposure to a specific antigen, so it's adapting to the antigen, right? Adaptive immunity. And it's characterized by two things. One, memory, and specificity. Okay, so obviously specificity is, do I target a specific antigen? Is it a specific protein on a virus or so forth? And memory, which is really key with vaccine, is does our immune system remember this organism and can it respond to it? And with vaccines, that is the goal, right? You expose your cells to a small isolated particle that doesn't have quite an effect, and now your immune system kind of remembers what it is and remembers how to have a quicker response to it. And this function, okay, the memory and, it's able to target specific antigen, and it's really dependent on two different lymphocytes. Lymphocytes are just a class of immune cells, okay? So B lymphocytes and T lymphocytes, you've probably heard of both. Okay, B lymphocytes help with antibody-mediated immunity, and as you expect from the name, antibodies lead it. T lymphocytes, or T cells, they are responsible for the cell-mediated immunity. Similar to antibody-mediated, you can tell this immunity is led by cells. Okay, and the organs that support this system, that is actually what makes up what we're going to discuss and what is known as the lymphoid or lymphatic system. This is just a quick diagram, all right, to show we do have some innate cells and strictly adaptive cells and phenotypes that kind of play a role with one another, but you will see there's one exact type of cell that crosses both barriers. That is the natural killer T cell. Okay, whereas we have our own classes with an innate immunity, it's important to know that only one of them comes across the fence. Okay, so the lymphoid system. All right, like I said, it's kind of our key little guy that helps to get this inflammation, this immunity to go. All right, and it consists of a couple things. One, the lymph plasma. It's also known as lymph, and it's just a clear fluid that's derived from our interstitial fluid, which is transported by what are known as lymphatic vessels. So there's vessels that help to transport this lymph, and of course we have the tissues and organs. Okay, so your lymph nodes, things like that, and they help to contain a lot of the cells or the lymphocytes that help to keep this system going and have the correct response. And of course, bone marrow, okay, because we need blood cells to help. Okay, some of them have their own response as well, but generation of the blood cells also helps with a healthy cardiovascular system, which helps with a more efficient spread of whatever immune response you have to have. All right. And it has lymphoid tissue. Okay, so lymphoid tissue is kind of like just a connective tissue, but the connective tissue has lymphocytes, which allows it to be counted as a lymphoid portion, I guess you could say, of the system. Now it has three primary functions, this entire system together. The lymph, the vessels, tissues, marrow, all that fun stuff. It's got its three functions are to drain any excess interstitial fluid, okay, and return it back to the bloodstream. Makes sense because we want to reuse it and recycle it. Transport dietary lipids and fat soluble vitamins, okay, so helping with the homeostasis, as well as we need some of this for the response and energy needed for inflammatory reactions. And of course, carry out immune responses, which is a big boy. All right, so we have all three of those, and that's what our lymphoid system helps to do. And you're like, okay, why does it matter if we're draining this fluid and returning it and doing all this other stuff? Well, if you don't have this control of drainage from the lymphatic, you can actually start to mess with a couple things, which will basically mess up our homeostasis. Our blood volume is going to be lost. Its balance with tissue fluid is going to be lost. So we're basically going to, if we're not draining it, we're throwing ourselves into this much more non-homeostatic portion. This is just kind of an idea. As you can see here, we have these nodes all throughout our body. You've probably, had your neck squeezed, to look at some of the nodes within your throat to see if there's any swelling. You can see thoracic ducts. Everybody here's a thoracics, obviously, but you can just see that overall that this lymphatic system kind of goes throughout our entire system. And more so, it has helped to be generated by these lymphatic vesicles. Again, you can see the pathway that it's not just, okay, it's coming out of one lymphatic organ, it's actually going all the way throughout the entire body, which helps carry the response needed. And now, as I said, we have to have some drainage and get rid of some of the lymph. And we'll cover this a little bit more. But as you can see, each what is known as a lymphatic duct has its own draining portion of the body, as you can see on the right here. So we have a right lymphatic duct and a thoracic duct. Now the area just drained by the right lymphatic duct, as you can see, is in the upper left-hand side of the body. Right, and basically the rest of the body is the area that's drained for the thoracic duct, and we'll talk more on exactly what that means. Okay, so as I said, lymph plasma. All right, so we need to actually make it, and we need to get it able to be circulated. Now, you guys sitting here going through this day, you have about 20 liters of fluid that's been filtered out of your capillaries and into your tissues. And about only 17 liters of that return to your bloodstream to kind of recycle. The remaining 3 liters of that fluid, however, those are what's going to go into the lymphatic vessels. That is what eventually generates the lymph plasma. All right, so by 24 hours, you've made 3 liters of lymph plasma. Now, this is not to get confused with blood plasma. Okay, so they differ, and it's mostly differed by their location and the fact that they have a lower protein concentration. Okay, so don't confuse lymph plasma with blood plasma. Okay. Now, we want to contain as much as possible, right? So the lymphatic vessels help to actually return any type of leaked plasma proteins to the circulation, and this helps to prevent any loss of osmotic pressure, helping of the flow by the pressure that is established. And so using this pressure, you'll see the cardiovascular system, we have a one-way system flow of lymph. Okay, starts in the blood, right? Once it's generated, it's in the blood, goes in the interstitial fluid. That then becomes lymph. The lymph then goes into a lymphatic vessels, and lymphatic vessels find its way to the veins, which veins is then where either the site is or where it's going to go back into circulation. And as I said, this flow is aided not just by cardiovascular, but also by the pressure and 2 other key parts. One being known as the skeletal muscle pump. So that actually is your contraction, right? Your contraction of skeletal muscle. And those contractions help to compress the vessels throughout your entire body and helps to basically give it more of a flow by increasing pressure. All right, and well as the respiratory pump. Okay, so similar to skeletal muscle pump, as you inhale, pressure is applied. So allowing the pressure to push whatever lymph and throughout the lymphatic vessels to where it needs to go. And it also helps to prevent any backflow, okay, within this one-way system by you exhaling, okay, because you're releasing and relieving the pressure to prevent any backflow. So both of those pumps play a key role in pushing all the way from blood to lymph to veins. Now, this is, again, just, quick little example of what exactly I mean. And what you see, let me get my laser pointer here. Okay, cool. This is just a depiction showing, of course, the heart, the cardiovascular system with the veins and the arteries. And of course, we're bringing in a lot of our lymphatic system. Okay, so the green here is lymphatic system. The rest is our cardiovascular systems. Okay, now, as we discussed, blood capillaries, okay, they bring in to the lymphatic capillaries, produce, the blood. We get the blood that goes into the heart, and so forth, so forth, so forth. It go through the way. And eventually the blood plasma hits a point where it gets filtered from the capillaries into the interstitial spaces. All right, so that is where we get interstitial fluids. We've gone from the heart all the way into blood plasma and into interstitial fluid. This is where your lymphatic capillaries, those actually absorb that interstitial fluid. All right, and that's when it becomes a lymph plasma. Okay, so heart to arteries, to blood plasma, the blood capillaries, and then we get to lymph plasma. All right, so then lymph plasma from there is carried all the way through capillaries within lymph nodes. It's filtering, removing any foreign substances. It's moving on forward. It's carrying the plasma more and more and more and more. And as you start to see, all these playing a role with continuing pressure. Okay, and keeping it going one way, such as the valve here, which basically happens exactly as you would think. It's like a one-way door to let the lymph go forth. And then eventually lymphatic vessels are hit. Okay, so we've gone from blood, out the arteries, into the blood capillaries, interstitial fluid, into lymph once it's a lymphatic, all the way through filtration and other processes in a one-way manner, hit those lymphatic vessels. Okay, so now the lymph plasma is in the lymphatic ducts or the vessels, continues on forth. And now when it gets to a lymphatic duct portion, okay, the plasma is into the junction of the jugular and subclavian veins of the cardiovascular system. Okay, so this allows it to pump back in, all right, hit again into the heart, pump on out, and pick, anything else up that is needed, or if it doesn't completely need to go all the way through those 3 liters, this is where the lymphatic vesicles would help with the delivery of whatever tissue it needs to go to. All right. So that was just kind of a quick on lymph plasma, how it's made, how it's moved. And now we're going to start talking about the actual tissues and vessels and ducts, and even a little bit on how they play a role in what we just saw. And what we, kind of got to in that process is that the lymphatic capillaries are very important. Okay, so they are very highly permeable, which makes sense because we need lymph to be pushed through and blood to be pushed through. Okay, now if you look at it at a cellular level, they contain endothelial cells. All right, and they are overlapping on one another, and kind of think of it as like a two-way door. Okay, so they overlap one another, but the door only opens one way going in. So think of it as a door that's there and it's opening. People are coming in. It's opening. The people are coming in. So within those capillaries, you have a permeable solution that's able to get through. The permeable solution flows through these overlapping endothelial cells that are more of a one-way valve, allowing it flowing further into the capillaries. Now, a key kind of thing with it is that this liquid is coming in with increased amount of pressure and speed. Okay, and so the body had to adjust with certain ways to control this. So we don't want a collapse of these super important capillaries, or else this is going to impact our entire immune system, our homeostasis. So it made what are known as anchoring filaments. So this, think of it as kind of like strap down, okay, to help prevent any collapse during tissue swelling. All right, so it helps it to keep extended as well as keep itself from kind of coming in on itself. And not all lymphatic capillaries are created equal. We have a lot of specialized ones. Most commonly, you'll see a couple specialized ones within the intestines called lacteals. These guys are what help to do that one function I mentioned, whereas they help to absorb dietary fats. All right, so they absorb dietary fats from whatever we've eaten, obviously, and they form basically a milky lymph that's rich in lipids. All right, so we have specialized lymphatic capillaries all throughout our body, but intestine I discussed because it really helps with one of the main functions. This is just a quick kind of example. Okay, so this is just a lymphatic capillary. All right. Now, as we saw before, right, we get some fluids that are coming in, commonly from blood, correct? It comes on in. We eventually get interstitial fluid. Interstitial fluid will eventually become lymph plasma. Okay, and as you can see with the lymph plasma coming up on the walls of this is where that endothelial one-way door overlapping allows the solution, black arrows, to come on in. Okay, and so like I said, everything here has some type of relationship with the cardiovascular system. And so this is just kind of a quick depiction of showing again how this simple little capillary structure functions. All right, so the green here is a lymphatic capillary, and as you can see, wrapped around it are arteries, okay, and veins. And what occurs is, as we saw, the lymphatic capillaries hit, and then what comes in is interstitial fluid turning into lymph plasma. This is then what allows it to continue on through the body with the combined pressure. But at the same time, there's overlap within the arteries as well as other portions of the cardiovascular system, which allows kind of more of a funnel back into the blood system. So it's not just like a lymphatic capillary over here is by itself. It is highly intertwined with our cardiovascular system, which makes a lot of sense because, they need blood to make it, but they also need blood to spread whatever response they need. Okay, lymphatic trunks and ducts. These guys have kind of a go and roll with one another to get an important thing done. And we have several trunks, okay? Lumbar, intestinal, bronchomediastinal, subclavian, jugular, all are considered important trunks, okay? And they assist with these ducts to help lymph be drained. So now if you remember that little diagram that we showed a little bit ago, and those two different colors that showed where exactly it was draining to, again, there is two important ducts that do this. Right lymphatic duct, so that was that upper right quadrant of that diagram we saw, and a thoracic duct, that was the entire rest of the body. Okay, and this all works together, the trunks and ducts, to help ensure that this lymph is drained and that it's returned to venous circulation. So this is just a quick diagram of where the trunks are located within the body, as well as kind of where the ducts are. Okay, so if you're looking forward within down below the neck, okay, we have our right jugular trunk, okay, which comes in together at the right subclavian trunk duct or trunk, okay. It goes a little further, again, right bronchomediastinal trunk, but on the left side, okay, we get our left jugular trunk, which leads into our thoracic duct. Okay, so if you remember, the thoracic duct works its way down and goes all the way into the rest of the body when it comes to draining. Okay, and now the bronchial mediastinal trunk has a left and right side. Okay, so both help do the same exact thing with drainage from the ducts. Okay, the thoracic duct. This is just a little closer, kind of look further at it on what exactly the right lymphatic duct is. Again, here it helps with the drainage in that one quadrant of the body, blood flow, lymphatic drainage. Okay, then the thoracic, which as you see it goes all the way down, right, and up. So this again helps with flow throughout the entire body and its association with the other trunks that help to complete it. Okay, so we haven't really got to the organs until now. All right, and we have some primary ones and we have some secondary ones. And the primary ones I'm sure you've all heard of. Okay, and why they are considered primary is because this is the site where those immune cells, those lymphocytes, become immunocompetent or basically active, ready to do the job. And there's two things that help to make them. Red bone marrow helps to make B cells. All right, and the thymus helps to make T cells become mature. So think of thymus, T cell. So of course, thymus is important, correct? So thymus located in the mediastinum. Okay, it's really prominent in childhood. All right, but it starts to slowly undergo this age-related involution as we start to age. And as we just talked, it's really key with maturing of T cells, getting to the point where they can do their job and they become active. So it is considered the site of T cell education. All right, so that is another terminology used for this maturation, whereas you are educating these T cells through positive selection, which we'll show what that means in a bit, to allow them to go out, do their job, recognize whatever they need to do, and to produce hormones that will then support future T cell development. Again, just a quick depiction, lungs, all right on the center, right below your jugular, okay, is where you'll see and feel your thymus, okay, until it goes about, a couple inches above the diaphragm, okay. And now this is just a quick little zoom in so you can see that in fact, we do have T cell populations, epithelial cells that make this thymic corpuscle, which helps with formation as well as production of thymic hormones that help to do a lot of the activation later down. Okay, so that was basically the primary one. All right, and of course, since we have a primary, we're going to have secondary. So we do have secondary lymph nodes. Interestingly enough, these you've probably heard a lot of too, but they're not considered, primary. They're considered secondary. So these are your lymph nodes, your spleen, and your lymphoid nodules. So MALT for short. So lymph nodes, they filter lymph plasma, okay, and it's allowing it to produce a site for immune activation. All right, structure is, it's encapsulated. It's got two different zones, so cortex and medulla, and the cortex contains the B cell nodules. All right, so where the B cells can be produced. The medulla contains macrophages and plasma cells, so that is the storage and the production of macrophages and plasma cells. Okay, so two different zones, each one has its own key role with the immune system. Now, since it has these B cells, obviously its job is to, one, activate lymphocytes. The S and that rest of it is, it helps with filter. Okay, so it filters to trap any pathogens that are in it. It filters as a way to slow the flow to enhance what is known as immune surveillance. But it also helps to produce antibodies. Now, antibodies, they come a lot into, obviously, our adaptive immunity, right? Because that's what helps us to build this immune memory. So the lymph node helps with producing antibodies, as well as producing B cells, macrophages, and plasma cells. So the flow in lymphoplasma and the lymph nodes, it goes in a pretty precise manner. Okay, so it goes through afferent lymphatic vessels. All right, that penetrates the surface of whatever node. All right, and it contains valves that open towards the center of the node. So again, like I said, one-way flow. It acts kind of like a door to open up and then close when needed to slow the lymphatic system. Okay. Now, once it gets in the node, the lymph plasma enters your sinuses. Okay, in a series of irregular channels, which contain a bunch of branching fibers, lymphocytes, macrophages. So you're starting to get into the immune cells, right? And in the process, when it gets there, it flows into the subcapsular sinus and immediately beneath the capsule through trabecular sinuses. Okay, you're starting to see the sinuses are getting a lot in there, right? This drains into one or two lymphatic vesicles. Okay, again, valves will open one way, away from the center of the lymph. And these vessels then emerge from one side of the lymph node at a slight depression, which is called the hilum. The hilum then is a site where blood vessels are entering, but also leave the node within the lymph node at the hilum. Now, when you look at it in a diagram, which probably helps explain it much more, you have your afferent lymphatic vessels. Okay, so we have some flow here, the lymph plasma. Okay, and now when it's in the node, because remember, after it passes through that one-way valve, it gets on into the node. This is where it starts to have these channels that are basically called sinuses. Okay, it's a series of irregular channels, and they contain a bunch of branching fibers, lymphocytes, macrophages, all these that you can see up here. And each of them have their own cells, which we discussed. Cells of the inner cortex, they have T cells, dendritic cells, cells of medulla, B cells, plasmacytes, macrophages. So what occurs is, at this point, your lymph has come in, it enters into basically these channels, and the channels flow through. Think of it as kind of like getting the message to the immune cells. Okay, and now eventually, once it does its jobs, it is activated, it's trained, it's got the signal going, it eventually has to flow out. Okay, so it's gone from one side of the lymph node all the way to the opposite side. Okay, this is when it goes through the hilum. Okay, so remember, hilum is just a divot where blood is flowing in, but also flowing out. Now, at the time of hits this hilum, it goes into what is known as afferent lymphatic vessels, which is basically just the exit. Okay, think of E, exit. All right, it goes out, the same type of method where the valve is opening the opposite direction to keep a one-way flow. As you'll start to see, this is a pretty big theme with this, is just it wants to go one way, and it has various ways to make sure it goes that way. And so once it hits this valve, it can then finally continue on through with the lymphatic vessels, carrying whatever cells that it's brought with it, whatever antibodies to help bring throughout the body. But the lymph node is not the only one that has a key role. Okay, and I'm sure you probably think lymph node is like super important because that's usually what everyone wants to check if you have this huge, terrible sickness and your immune response is occurring. Do you have small lymph nodes? I'm sure you've heard. But there's more than just that. Okay, the spleen, for instance, it's actually the largest lymph node organ, and it helps filter blood. Okay, so we've talked about lymph throughout everything. Spleen filters blood, not the lymph. Okay, and it has two different, I guess you can call them zones. All right, white and red pulp. White is where the immune function is. Okay, it's rich in lymphocytes. Okay, so again, lymphocytes we'd expect. Okay, T cells, B cells, other immune cells. So that's the white pulp is where it's rich. This is where the immune response comes. Now the red pulp, blood, red, it's going to remove old or damaged red blood cells. And it actually helps to store platelets. Okay, so each role has its own responsibility. And since it has different functions, each of it has its own structure. Okay, so obviously, when you think of a spleen, you probably think of this structure. Okay, the spleen also has ileums. Okay, as well as arteries, because obviously they're getting through some flow, and other lymphoplasma and lymphatic vessels. Okay, and now looking at the spleen, like I said, they have different types of structure, all depending on what they need to do for their region. All right, so white pulp, for instance, you can see it's quite thick. Okay, that is because there are so many lymphocytes that are just kind of squeezed into one area for an immune response. So they kind of are also a little sticky depending on the cell. So they kind of grab onto one another, you can say. And red pulp, where you need a lot more space, a lot more artery kind of introduction to allow the blood flow and the release of any damage. So each pulp has its own structural change when it comes to its function. But you will also see that in the white pulp, we also have arteries. Makes sense. Once we make the cells, we got to get them somewhere. So having arteries protrude through the white pulp helps as well. Okay, so lymphoid nodules are MALT. They are unencapsulated. Okay, so remember, lymph nodes were encapsulated. These guys are unencapsulated. And they're found actually in the mucosal linings. So this is like your tonsils, your appendix. They are kind of like a gate, okay? And they're applying the first defense. So they're commonly found at places where pathogens will most likely show up first. Okay, so like tonsils, you're breathing in some type of airborne virus, eating something that has a microorganism on, for an example. They're there at those first entry points that pathogens may be exposed. Okay, so those are a quick summary of what organs are involved in the lymphatic system. Okay, and as with everything, it has to be developed in a precise manner when we're being born. And by the 5th week of embryonic life, our lymphoid tissues are developed. All right. And again, we all derive from, or portions derive from different types of tissues, such as our lymphatic vessels. Those are mesoderm-derived lymph sacs. Our thymus develops from a pharyngeal patch. The spleen forms from mesenchyma and the dorsal mesentery. But if you look at it, okay, like I said, 5th week development. All right, so we got some structures, and we have our jugular lymph sac. Okay, and as you can see, the lymphatic tissue or the vesicles are already kind of starting to form, as well as internal veins, which again are already starting to associate with one another. Okay, and at these specific regions, we have a jugular lymph sac, a retroperitoneal lymph sac, and a posterior lymph sac at this 5th week of embryonic life. Okay, and each one of these are then what develop into our thymus, our spleen, and so forth. Okay, so we got an idea on exactly what is involved in these. All right. And as I said in the way back beginning, is our innate immunity it has its own first line defense, and that is your skin, chemicals. You actually have good parts of your body are actually acidic, which help with chemical defenses. Lysozymes. You also have mechanical removal, so like shedding, fluid flow, keeping them from either embedding into the cells or releasing what they need to cause harm. All right, so when it comes to innate immunity, those three help with acting as the first line of defense. And so it may not be that they are technically there to attack and kill, but these defenses are there to prevent entry and colonization. So its main goal is if it's not getting in or if it's not able to grow or colonize, then I don't have an infection. So it's its first line of defense to prevent this from occurring. Now this is just a quick summary of some examples of what exactly our first line of defense entails. As I said, we have skin, physical barrier. We have membranes at the many entrances of our body. Now, it's not as effective as if we had intact skin, but it can also act as a physical or sometimes even more chemical when it comes to mucus to keep them out from entering. Our hairs, okay, so those are quite important. Nobody likes hairs coming out of their nose, right? Those are actually quite important because they help to either catch or filter out the dust within our nose or anything else we may be inhaling. All right, so cilia, those all usually work together with mucus. All right, they trap and remove any microbes that are from your upper respiratory tract. And there's tons of other. All right, so lacrimal apparatus, that's your tear ducts, right? So if you have something in your eye, it helps you to dilute it. Your saliva and your urine, saliva helps with, mucous membranes. Urine, obviously, we excrete what microbes may have. Vomiting, it's actually considered a first line of defense. Okay, so obviously, if you ate something bad, you have, salmonella risk. Your body's going to try and vomit it up to get it out of the system. So those are all physical factors. Now, chemical factors, a lot of that comes from like enzymatic or acidic. Okay, like gastric juices. It's acidic, but also enzymatic. Lysosomes, those are enzymatic. They, are in your tears, they're in your saliva, they're in your nasal secretions, they're in a lot of the fluids that are surrounding your tissues. Sebum forms a protective acidic film over skin surfacing to inhibit the growth of many microbes, as well as vaginal secretions. Okay, the acidity again discourages bacterial growth. So a lot of it is more of a enzymatic and acidic factor when it comes to chemical factors. So that was the first line of defense. This is the second line of defense of innate immunity. This is your internal defenses. Antimicrobial substances and natural killer cells as well as phagocytes play a key role. Now, our antimicrobial interference. This helps to inhibit any type of viral replication. As you may know from viruses, they hijack our system, okay, to produce the DNA that they cannot produce or the RNA that they cannot produce. These work together to kind of help and give another check to that to prevent them from finishing the replication. So forth, they can't survive, they can't divide. Eventually we get better. Complement. This is a complement system. Then it is a system that is there to actually enhance a lot of immune reactions. We'll talk a little bit about the complement system later. Iron binding proteins. All right, a lot of microbes need iron to survive. So these proteins go and they kind of act like a sponge. All right, and they deplete some iron in the surrounding environment of the infection, which allows the microbes to not survive and not thrive because they don't have enough iron to do so. And of course, straight peptides that will direct kind of like a fill or a flag, putting into whatever microbes and kind of getting it as a homing beacon to be attacked. Now the cell side, okay, because two cells play a key role when it comes to this type of immunity. Okay, natural killer cells and phagocytes. Natural killer cells, they destroy and detect an infected or abnormal cell. These guys play a pretty key role when it comes to cancer as well, okay? Because, when we're infected, when we have cancer, our cells become abnormal. They're not usually doing their normal structure, their normal function. So they are there to basically act like the security guard and check and see what's wrong. Now, phagocytes, you probably heard of phagocytosis, right? It's exactly what they do. Right? They ingest and destroy pathogens via that process. And the process that is chemotaxis, adherence, ingestion, digestion, and then eventually killing. Okay. Now, if you don't remember, I'll cover phagocytosis really quick. Okay. So, like I said, it's got 5 phases. First one is chemotaxis. Okay, so it is just when there's a chemically stimulated movement of a phagocytes to a site of a damage. So it detects there's something over there that is causing an infection or damage. I need to go there. So that allows this phagocytes to get there. All right. Once it gets there, it then undergoes adherence, and exactly how it sounds. It attaches the phagocyte to the microbe or whatever other weird foreign material that it needs to deal with, because you can't really do your job if you're floating away from it or not able to get a hold of it. Now, this is when it starts to be probably the phagocytosis you think of, and that's when ingestion starts. So that is just when the plasma membrane of those cells, they extend to make little projections. Those pseudopods you've probably heard of, and then again to fuse together and allowing the cell to engulf or take in whatever it wrapped around itself. Okay, and this, once they fuse, okay, the ingestion is done and it's fused, it becomes a phagosome. Okay, so it's gone from pseudopod to phagosome. And then we get digestion, and it's exactly as you think. The digestion is just, it comes in and it uses enzymes. Now, the most common way it does this is it fuses with the lysosomes in our cells, because remember, lysosomes have a lot of enzymes in them, especially ones that help to break down DNA and proteins. Definitely protein. They got a lot of protein. And so those lysosomes and those phagosomes fuse together, leads to breakdown of proteins, cell walls, all by enzymatic cleavage. And eventually that literally leads to just the killing portion. All right, so the killing portion is finally done. Whatever was inside there has now been destroyed. And usually the cell will then just plop it out. Okay, just shoots it out the other side. It's kind of almost like it ate and then passed. Okay, so that is just a quick idea of what these phagocytes and phagocytosis is really key to do in our innate second line of defense. This is just another graph similar to what we saw with the first line of defense to cover some of the key things. Okay, so again, we talked interferons, all 4 viral infections. Okay, then there's a viral infection. Not only will they help to prevent the virus from replicating and thriving, but it also acts kind of like as a gate to prevent any type of viral infection in uninfected host cells. Complement system, okay, promotes basically almost everything inflammation wise, like phagocytosis, cytolysis, a lot of stuff that contributes to inflammation. There's iron binding proteins, which lead to inhibition of bacterial growth from removing available iron. Antimicrobial proteins, okay, we talked about them previously, but they also function here by impacting other cells that we didn't talk about, known as dendritic and mast cells. Okay, and these are another form of an immune cell that these proteins can help to bring to and kind of do their role of destruction of any type of microbial organism that's kind of found its way in. Natural killer cells. All right, so like I said, they help to detect if there is something wrong with cells, and they will kill those infected target cells by releasing what are known as granules. Okay, so these are granzymes, perforin. Basically, it's what helps with bringing the phagocytes towards where they need to go. And once they're there, the phagocytes ingest and destroy whatever matter. And now this is considered an internal defense when we talk about inflammation as a whole. Okay, so inflammation is actually a second line of defense, internal defense. And as such, it doesn't just refer to as, you're getting a virus or a cold. It actually refers to as tissue repair when it comes to this type of internal defense. And lastly, fever. Fever is considered a form of internal defenses, and intensifies a lot of these other guys, interferons, speeding up body reactions that may aid repair, producing more energy, as well as building blocks of tissue to help with repair. It's why almost every inflammatory sickness you have is associated with a fever. So, we talked about it right there. And if any of you have ever heard about inflammation before, you've probably heard of PRISH. Okay, so inflammation is a non-specific protective response to tissue damage, okay, or swelling, but in this instance, we're going to talk about tissue damage, okay, whether it be like a cut, a bruise, things like that. But most commonly, the signs of inflammation with this are known as PRISH. All right, pain, redness, immobility, swelling, and heat. Okay, this is why if you ever cut your finger, for instance, it will hurt really bad. Okay, and then it will start to turn red around the cut. You will start to have issues with mobility with it because of the cut. It will start to swell, and then you'll kind of start to feel some heat coming off of it. Okay, and so this all occurs because it's undergoing inflammation, and it occurs in 3 stages. Vasodilation, okay, that's where we increase in diameter, and then increase permeability all at the same time. Immigration of phagocytes, and then tissue repair. Okay, the stages happen in 3 in that order. Now, it has to do a couple other things at the same time. It's not just for tissue damage, for the inflammation to come in, do those 3. It actually has to find a way to isolate the damage in the aspect of if there's any pathogens that may be coming into such a wound, and they can heal or initiate or stop the healing, for instance. So not only is inflammation there just to help with tissue repair, but it's there to also kind of act as a border to prevent any pathogen from causing a further infection. So vasodilation. Okay, so vasodilation is basically changes within the dilation of capillaries, all right, to help bring phagocytes to whatever spot of injury has occurred. Okay, and now the substances that help it, there's basically 4 or 5 big ones. Okay, histamine is one. So this is commonly used for response in injury. It recruits mast cells, basophils, platelets, and basically all to the site of where the vasodilation is needed. Okay, and even further is it brings neutrophils and macrophages there to help with any type of infection that may be incurred, and they further release histamine too to kind of ramp it up. Kinins are polypeptides that are formed in blood. Okay, so these are actually inactive precursors to kinins. These induce vasodilation, and they also increase the permeability, and they help to kind of act as a homing beacon to where those phagocytes need to go. So that is some type of chemototic agent that they will use to then do chemotaxis. Prostaglandins, these are actually released by damaged cells. So when cells become damaged, say their walls rupture, or they just have a leak in their membrane, what happens is cells will release these. Okay, and not only can it act as a chemotactic agent by itself, but it actually can intensify all the effects of the other 2 I just mentioned. Okay, which if they're intensifying the effects, we're going to lead to an increased kind of recruitment of other immune cells as well as phagocytes. All right, and this itself actually helps the phagocytes come through capillary walls, which we'll see in a diagram exactly what I mean by that. All right, so that's comes through the capillary walls to get to the damaged tissue. Leukotrienes are produced by basophils and mast cells. Okay, so these are specifically made by of basically a type of immune cell or leukocyte. These are there to increase the permeability. All right, so yes, we can, these phagocytes can leave the capillary walls, but it needs to increase this permeability of other things to come across the walls, such as the cells and others. And it also functions as an adherence, because if you remember, on that adherence part, they needed something to grab onto to stay in the location of whatever pathogen or damage they need to fix. So these guys can act as an adherence for them. Now, lastly, the complement is kind of its own thing. It's called the complement system, but it does impact this stuff. Okay, so the complement system can stimulate the histamine release. It attracts neutrophils by the chemotaxis. It helps to promote the phagocytosis, which is basically the endpoint of all of these together. So the complement system acts kind of as a ramp up system to keep it going and keep it going stronger. But the big takeaway here, okay, is that the prostaglandins, okay, they basically help with immigration of phagocytes. Leukotrienes help with permeabilities and adherence. Kinines, they help with permeability as well as chemotaxis. And histamine kind of does it all. It's more of a, I guess you can say, a chemotaxis agent for neutrophils and macrophages. Okay, and the complement system just helps with every single thing. Now, this is what I mean when I was talking about it. Okay, so phagocytes have to migrate from blood site to tissue injury. Okay, so all that occurs is vasodilation. Okay, whatever release of the histamines or whatever else we just talked about occurs, and this basically extends in diameter. The walls of the capillary walls become more permeable. Okay, so phagocytes will follow whatever signal they're getting, the chemotaxis. They will protrude themselves through the capillary walls. This is your immigration phase. Okay, and it can either be one phagocyte or two phagocytes or several different type of ones. And either way, what happens is they get there, all right, by chemotaxis, and they do, phagocytosis. They bring it in, break it down, and they kill it. Okay, if there's any pathogen or remake, and it's, a lot of this is there to help anything that may delay any type of healing, such as killing, or not killing, but breaking down dead cells, who they themselves, if they're dead or ruptured, can lead to toxic chemicals being released and stopping the healing. All right, so that's why you do a lot of your wound cleaning, not just the pathogens, but a lot of the proteins and enzymes, like the ones that would be used in phagocytosis. They can damage other cells if it just happens to release by a rupture. Okay, so that was all innate immunity. Now, adaptive immunity, okay, innate was an immediate and non-specific defense. Okay, so remember, innate immunity, we have it right as we're born. Adaptive immunity is highly specialized. Okay, and it develops over time. Okay, so as we age, our adaptive immunity starts to get more and more trained. Okay, and it gets more able to target whatever specific foreign pathogen or antigen, okay, a foreign substance known as an antigen, to then have a response to be able to take it down quicker, okay, and efficiently. Now, there are the two defining characteristics, okay, that distinguish adaptive immunity from innate immunity, and you should remember them: specificity and memory. Okay, obviously, specificity is for its ability to discriminate, if this is an antigen or if this is a protein that I made, or if this is one antigen versus the other antigen. So it's the ability to distinguish antigens from self and other antigens from others. Memory, okay, they need to have a memory so that if they are exposed to another virus, say like the flu virus, then it knows, I've seen this before. I know what response I need to have to destroy the virus and to prevent the infection from going. And so that is memory. Then it's a faster way for the cells to have a quicker response to the exposure of the same antigen it's already seen before. Again, this is where it comes from vaccines. This here is ways to acquire adaptive immunity. Okay, like drinking water out of the water hose, right? Kind of. So, naturally acquired active immunity. Okay, that's just exposure to microbes and antigens over time. Okay, leading to whatever recognition B cells and T cells need to do, which actually leads to formation of plasmacytes to secrete special antibodies, which lead to any type of special activation of T and B cells, which we'll see exactly what I mean by that. So that's literally just us growing up, being exposed, getting sick when we're kids, and our body just naturally kind of acquires an active immunity. Naturally acquired passive is IgG antibodies. Those get transferred from mother to fetus, okay, across the placenta. IgA antibodies also get transferred, however, that's more from baby, for milk during breastfeeding for the babies, okay? So this is kind of why some pregnant women, they suggest to get vaccines while you're pregnant, okay, especially if contradicting blood types, O negative, O minus, because the body can kind of have a response to the fetus. Um, they have them because these antibodies are able to pass to a baby and a possible passive immunity, and so then you won't have to expose the baby to the vaccine itself. Now, artificially acquired active immunity, that is the vaccination side of things. Okay, and so memory cells being produced and all, antigens being identified previous. Artificially acquired passive immunity is sometimes we are injected with immunoglobulins or antibodies that may help with whatever disorder, disease we may have, or autoimmune diseases. So we can obtain what is known as active or passive immunity in natural and artificial ways. And as I mentioned, yes, we have antibodies when it comes to adaptive immune responses, but it's very heavily cell. Okay, it's more of a cell mediated side of things. And we have the lymphocytes that we've talked about before. Those are your T and B cells. But we also bring in the antigen presenting cells. So those were your macrophages that I discussed. Your B cells are there to as well present antigen. So this, what the name is telling you and what it means is that these are cells that go and find whatever, bad thing is there, and they present the antigen that they found to whatever lymphocyte needs a response. So B cells presents it to helper T cells, which are a classification of T cells. Dendritic cells, they would present it to T cells and B cells. All right, macrophages, they present it to T cells. Okay, and they can actually lead to cytokine secretion, which we'll get on to what those are as well. Now, I talked about helper T cells. This is classification of lymphocytes. So they just work together with B cells. Okay, so work together with B cells, and they actually release some of those cytokines that I'm going to talk about, which help the inflammation system continue on. Now, outside of just a T cell and a helper T cell, we have cytotoxic T cells and memory T cells. Cytotoxic T cells, they go and attack. All right, they are the cells that will go and kill whatever host target cell is there. They release granzymes, inducing cell death. They reduce cytolysis. They just go and they do the dirty work. Okay, whereas memory T cells, these guys, they don't leave lymphoid tissue. Okay, they remain in lymphoid tissue, and they'll recognize any original invading antigens even years after first encounter. So that is kind of where your memory function comes from. B cells, they can actually become lymphocytes, as they are lymphocytes, but they can do a similar function in this manner, is where they become antibody-producing plasmacytes, so cells that produce antibodies. Same with plasma cells. Okay, and just like T cells, okay, we have memory B cells. All right, so this is just a divided down descendant of a B cell that's been exposed to an immune response, and it is able to respond rapidly to whatever same antigen in the future. So as you can see in the last slide is, again, this is very cellular mediated. And the lymphocytes you saw, they just weren't B cells and T cells as a whole anymore. They were tons of other different types of cells, right? And so while adaptive immunity does depend on two major types of cells, which are the B and T cells, they kind of become activated and matured into other types of those cells. Okay, and they all emerge from a multipotent stem cell that's found in red bone marrow. All right, and they each go through a type of maturing, all right, maturation. B cells complete their development within the red bone marrow, all right, and it continues all the way throughout our life. Now, T cells, they have these things known as pre-T cells, and they migrate from bone marrow all the way to the thymus. Now, in the thymus, that is where the T cell maturing occurs, and it becomes immunocompetent, so able to do its response. However, most T cells form before our puberty. All right, and maturation, however, continues throughout life. So we have the T cells, they're formed. It's just they aren't mature, I guess you can say, or turned into whatever immune competent form of T cell that we need. So, okay, so B cell, red bone marrow, goes throughout life. T cell finishes in the thymus, and it's only matured throughout our life. Okay. Now, these guys do something, like I said, the whole goal is to get them to be immune competent, right? So, what is immune competence? All right, well, what it is kind of what I said. They need to be able to be prepared to fight whatever immune inflammation is occurring. So before they leave their primary lymphoid organs, those cells, they have to develop it and gain the ability to recognize whatever specific antigen they may need. Okay, and how they go through with that is that they get insertions of receptors that are specific for whatever antigen the body is currently fighting with. That's what helps to kind of get it to its point where it can actually do its response. So, immunocompetence, kind of already talked about that one a little bit. Okay, so like I said, it's all before they leave the primary lymphoid organs. Okay, so remember, this is all in the primary. That's where the B cells and T cells need to develop this. And they gain that ability to recognize, and it's all by insertion of antigen receptors into their plasma membranes. So if you remember from way back, this all occurs through change of gene expression by signaling, which leads to promotion of protein formation within the endoplasmic reticulum. And the endoplasmic reticulum, which is generating a protein and transports it to the plasma membrane, all according to whatever response or other cell interaction occurred to make it ready to roll and ready to have what it needs. So, like I said, okay, it's no longer just a T cell, it's different classes, and there's two major classes that come from the thymus. So remember, when they're in the thymus, they are T cells. When they come out of the thymus, they are classified into two different types. Okay, before they get to the thymus, they are pre-T cells. I know it's a whole lot of T cell acronyms you got to remember. So, helper T cells. These are commonly referred to as CD4 T cells. And why they're called that is because there's a specific surface protein they express called CD4. And so a lot of researchers and clinicians use that as a common marker. So they know if you have, a swelled amount of numbers of CD4 T cells or a low number. Okay, and these guys, as the name kind of says, they help. And by helping, they coordinate the immune responses, and they coordinate it by letting the activation of other cells occur. So they will send off signals to activate B cells, to activate the other type of T cells, to activate macrophages, phagocytes. They are there to help activate, whether it be by releasing something or actually they come and they bind to one another and lead to some form of cellular communication that allows the other ones to become active. And it is essential for both branches of adaptive immunity. Okay, now on the flip side, much more of a stronger, I guess you can say, fighter are cytotoxic T cells. So these guys, similar to helper T cells, have their own surface protein. This is known as CD8. So the CD8 T cells, similar to CD4, the CD8s become a marker for researchers and clinicians to know which is which. These ones are more of the battle hungry. Okay, so they will go out and they will directly kill an infected or abnormal or foreign cells. Okay, it's key there is it's directly. They either release some form of an enzyme that will lead to the breakdown of the plasma membrane. Sometimes they can even release certain chemicals that will go into the infected cells and break down the DNA to break it down with inside. Okay, but these guys, all in all, are just they're the badasses that are central to all cell-mediated immunity. Now, I kind of said back there that there was two interconnected forms. So if you remember, it was cell-mediated and antibody-mediated. Okay, so as we just said, cell-mediated cytotoxic T cells are involved. And it's all for target cells that contain antigens. So antigens that are able to be recognized by the immune competent T cells or other cells. And now each one of these two, okay, they have their own effective and not so much effective abilities. Okay, so cell-mediated is very effective against any type of intracellular pathogen, so like viruses or parasites. It's really helpful with cancer cells. Okay, unfortunately, it's really effective against transplanted tissues, which is why there is a lot of precaution into controlling of the immune system, and that's why there has to be such a perfect match of transplanted tissues, because you don't want to get transplanted tissue that your body will recognize as foreign material and just go ahead and destroy it. So a lot of times this is why they will take some type of immune medication to kind of dampen down the immune system. And it also involves cells that attack other cells. Okay, so obviously that's key because it's cell mediated, and the next one is not so much cell mediated. Antibody mediated. This is also known as humoral immunity, but it will, instead of involve mostly cytotoxic T cells, it's going to involve B cells. Specifically B cells that differentiate into plasmocytes. Why they want to differentiate into that is the plasmocytes have a certain ability, and its ability is it can secrete antibodies, also known as immunoglobulins. You might have heard them that way. And if you remember anything, antibodies can bind to antigens, antibodies, okay, within bodily fluids. And for these guys, they're most effective against extracellular pathogens, so things outside of a cell. All right, and this is a specific type of immunity. The humoral immunity is so just unique because those antibodies don't just go in lymph or blood. It goes through both blood plasma and lymph plasma. This, I just have a little sheet for you guys for broken down. Okay, main cells and cell-mediated cytotoxic T cells, antibody-mediated B cells, plasmacytes. Targets are infected or abnormal cells and cell-mediated, and antibody-mediated free antigens. Okay, so just antigens that are flowing in our system. Pathogens for cell-mediated, intracellular, antibody-mediated, extracellular. Key molecules to help in cell-mediated are those preferins and granzymes. Those are the guys that the cytotoxic T cell secretes to help break down the cell wall or break down protein and kill the cells basically that are attacking. Antibody-mediated, obviously antibodies. But a key thing to remember is both of these require helper T cells. Okay, so it's not just one or the other. They both will need helper T cells or CD4 T cells to keep it along. Okay, and it's not just one or the other. All right, so don't think like, oh man, I have this infection. It's definitely just all my cell-mediated side of immunity going. And most, it's actually both. They actually operate simultaneously, and they work together to basically eliminate any antigens that are in circulation or any antigens that are specifically in tissue. Okay, so both of them will come together. It's not just one or the other. Clonal selection. So, only a small number of lymphocytes possess receptors that can recognize a specific antigen. Okay, I know I just talked about how estrogen, in the plasma reticulum makes them, it brings them up to the plasma membrane, but only a small number of them possess receptors that can recognize a specific one. And when an antigen binds to whatever matching receptor that's on that lymphocyte occurs, it undergoes what is known as clonal selection. All right, and it's a process in which that lymphocyte it proliferates or divides, differentiates into specialized cells, and it produces a clone of identical cells with the same antigen receptor specificity. Okay, so hence its name clonal clone. It's literally going to go and have this clonal production of its own self, and its selection occurred by the antigen that bound to it. All right, and this most commonly occurs in secondary lymphoid structures, so that's like your lymph nodes, the spleen, the malt. But B cells and T cells both, have to go through this process. Okay, it's not just one has to do it and the other one doesn't because they all have the receptor. And they both have to go through this type of selection. Okay, so like we mentioned, they all rise from those pluripotent stem cells. Okay, red bone marrow. We know that B cells go straight in. Pre-T cells go into the thymus. There's where they would mature into B and mature T cells. Okay, and now they'll develop in the lymphoid tissue. Okay, and they become activated in the secondary lymphoid organs as well as tissues if need be. Okay, and once activated, each type of lymphocyte forms a clone of cells that can recognize specific antigen. Okay, so these guys come in. They have the CD8 protein, so they develop in the cytotoxic T cells. They have the CD4 protein, so they become T helper cells. The mature B cells come in. They stay B cells. Okay, each having their own form of an antigen receptor. Okay, now remember, T cells help each of them. So the helper T cells come in, they can become what is known as active helper T cells. So those active helper T cells, they will come over and they help the cytotoxic T cells, but they can also come and help the B cells. And this interaction with both of them, whether it's indirect binding, leads to an actual activation of a cytotoxic T cell or an activation of the B cell. So, what goes forth is it goes out in a cell-mediated fashion for the T cells, and once it's out and it's active, it can go forth and attack whatever, tissue or invading pathogen it needs. B cells, if they go out, they get a response. What happens is they become plasmacytes. Okay, so the activation of the B cell gets a response, it comes plasmacytes. So in the process, it's basically, think of it as the way it's generating a membrane receptor. Okay, so hydrophobic, hydrophilic, right? It gets rid of that part that would bound into the membrane that you would see up here. Okay, so that part that is intracellular across the membrane just gets cut off. It doesn't get made. So in the process, the antibody or the receptor that would have been on this plasmacyte is now free floating. Okay, and then those free floating antibodies are then allowed to bind to whatever inactive antigens or go off into bodily fluids to keep itself going. All right, but the key thing here is all of this can occur unless there is some form of colonial selection up here. Okay, where they are able to basically replicate themselves with that specific antigen receptor to have a specific response. Okay, so this method of colonial selection, it's going to produce two major cell populations. Okay, those are your effector cells and your memory cells. So the effector cells were like the CD8 toxic cells or the plasmacytes. Those guys go and they actively eliminate or attack the antigen. So they are having an effect, or an effector cell. So like I said, cytotoxic T cells, plasmacytes, and actually activated helper T cells are considered effector cells as well. Most of these guys don't make it past the immune response. Okay, they will die after the immune response occurs. All right, so they're very short-lived. Memory cells on the other side, those guys are long-lived. And they may not be so active. They may not be out there fighting so actively. They've become more of a dormant type cell. Okay, until there's any form of re-exposure to whatever specific antigen. And because of their dormancy and their ability to remember, that is what allows us, if we have some form of re-exposure, to have a faster and stronger immune response if we're re-exposed to it. Okay, so I've been talking about antigens and all that fun stuff, but antigens also have their own kind of properties. Okay, now antigens have the two essential properties that are immunogenicity and reactivity. Immunogenicity is its ability to stimulate an immune response. Reactivity is its ability to bind to antibodies or antigen receptors. Okay, and now small regions of these antigens will actually trigger immune responses. Okay, so a small region on an antigen that actually does that, those are referred to as epitopes. Okay, or sometimes antigenic determinants, I refer to them as epitopes. Okay, so if there's that tiny region of an antigen that gets an immune response, it's called an epitope. And a single antigen can have more than one epitope. Okay, so it can actually be recognized by different lymphocytes because of this. All right, so a piece of an epitope on one may be more recognized by a cytotoxic T cell, or may be more strongly recognizable by an antibody. Okay, so a single antigen can have multiple of them, which can stem to different cells or different types of the immunity response. Now, they also have other natures, such as chemical natures. All right, so most of them are your proteins, your glycoproteins, your large sugars, lipoproteins, so fats with proteins. But they are almost all small molecules. Okay, they lack immunogenicity, but they can bind receptors. Okay, those are referred to as haptens. Haptens become immunogenic only when they attach to a larger carrier molecules. All right, so this is how we get some of our drug allergies and some reactions to poison ivy. Now, this kind of response, and dependent on antigen receptors and epitopes, we have our advantages when it comes to that, and it's because we have a diversity of antigen receptors. Okay, our immune system can recognize over 1 billion different epitopes. Okay, which gives us a pretty well dealt hand when it comes to fighting back on some type of infection, and it's all done by genetic recombination. Okay, so when we have gene segments that are encoding antigen receptors, and they get shuffled or rearranged during lymphocyte development. So if you remember your exons and introns and some of them being left behind and changing how the genes exactly are expressed and so forth, and changing how the proteins are structured, they can have this recombination of the same gene that gives us a different type of receptor. Okay. However, each B and T cell will express a unique receptor that enables recognition of a ton of antigen diversity. Okay. Now, the major histocompatibility complex, also known as MHC, I'll call it that because I want to save some breath. The MHC molecules, these are actually self-antigens. Okay, so they're not like we've discussed prior, where they're just antigens and epitopes and all that fun stuff that are just coming from an extracellular environment. These ones we make. Okay, and why we have them is because we need our cells to be able to say, "This is me. Wait a minute, this is not me." So it's a way for our T cells, since they're one of the more upfront, hands-on cells, to distinguish self from one another. All right. Sometimes you hear it as a human leukocyte antigens. I usually refer to it as MHC. Now, this MHC is unique to each individual, of course, except identical twins, because they come with the same genetic material. And we don't have just one MHC. Okay, we have two different types. MHC1 is found on all nucleated cells. Okay, and it helps to present endogenous antigens. MHC2 is found on antigen-presenting cells, and it presents exogenous antigens. So each one has its own role, one endogenous, two exogenous. Okay. Now, a fun fact about MHC is there is some studies that are suggesting that genetic variation in the MHC, as well as a couple other portions of the immune system, is actually what attracts us to specific mates, which is commonly why you may see one parent is sicker while the other one is totally fine. It's because there's some form of unknown pheromonic response with the immune system that is basically attracting one mate to the other that may have a stronger immune system. Fun fact there, mostly because I'm sick and my wife's not right now. Now, the MHC molecules really come into play when antigen presentation is being done. Okay, so T cells cannot recognize free antigens. They have to be processed and presented with these MHCs to whatever cells they are looking for. Okay, and now exogenous and endogenous antigens have similar but different ways of being presented. So exogenous antigens, as the name tells, it's found outside of cells. It's processed by what are known as antigen presenting cells. So this is your dendritic, your macrophages, your B cells, and specifically it's presented with MHC2. And it's there to activate helper T cells. Looking at the process of exogenous antigens being, processed and presented, the first thing that needs to happen is APCs need to first ingest the exogenous antigens. That's normally done by phagocytosis, but also endocytosis. Now, once they get into a phagosome or endosome, depending on whatever process they decide to go with, it then is digested and the enzymes go in and they start to break it down. They split the large antigens into short fragments, for this instance, short peptide fragments. And as this is happening, APC begins to synthesize the MHC2 molecules, because remember, MHC2 is here for exogenous antigens. And once it's synthesized, the MHC2 molecules are packaged into vesicles, and the vesicles containing the antigen fragments and the MHC2 fuse together. After fusion, the two types of vesicles and antigen peptide fragments bind to MHC2. So this all combined and interacting, it contains what is known as an antigen MHC2 complex, which then undergoes exocytosis. Okay, and it actually becomes inserted into the plasma membrane. It's kind of acting like a transmembrane protein. Okay, so after processing of the antigen presenting cell is now able to migrate to lymphatic tissue and presents that antigen to the T cell, which then allows it to become mature and get ready to do whatever fighting response it needs to do. Now, to see that kind of as a diagram, all right, we have exogenous antigen comes on in, phagocytosis or endocytosis, it forms a phagosome or endosome. Now, at this point, this is when we have digestion of antigens into peptide fragments. Right? So we use enzymes like proteases, stuff like that. But at the same time, we have the synthesis of MHC2 molecules, and that's happening in the endoplasmic reticulum. Just as a lot of transmembrane proteins, when this is occurring, it gets passage into a vesicle, all occurring at the same time as the antigen being digested. Now, once that digestion is done and the MHC2 is fully formed and in its vesicle, they will eventually fuse together. All right, and in the process of fusing, these antigen peptide fragments, they bind to the MHC2 molecules. The vesicles will then undergo your exocytosis, so getting, instead of taking it in, exiting out, and it's the antigen MHC2 complex that is formed and inserted into the plasma membrane. All right, and that's now when it can go and do its job with whatever lymphatic tissue it's designated to go to. Now, on the flip side of endogenous antigens, endogenous, they're on inside infected or abnormal cells. So this is actually processed by the cell itself, and it's presented with MHC1 this time. And this is there to do its process and activate cytotoxic T cells, or the warriors of the front. Now, processing of endogenous antigens is very similar to exogenous. It's actually shorter, but what occurs when this is happening is that if you have an infected cell, protein digesting enzymes, so very similar to what we saw in exogenous, it starts to split endogenous antigens into short peptide fragments. So right now it's looking very similar. It's just there's no phagocytosis involved. And similar to previous, at the same time that's occurring, MHC1 this time is being synthesized at the endoplasmic reticulum, putting it into a vesicle. Okay, now the antigen peptide fragments enters the ER, and then within the ER it binds to an MHC1 molecule. Okay, so the difference here now is instead of two vesicles forming together to form that complex, the peptide fragments of the antigen are going into the ER where MHC1 is being produced. Now, once it's in the ER, just as previously, the antigen and MHC1 come together, they form a complex. And those complexes are packaged in the vesicles. And just as previously, vesicles come off, they undergo exocytosis, and then the complex becomes within the plasma membrane. And now it can go forth into the system and kind of find that cell that it needs to find. Putting this kind of into more of a diagram form, okay, you have your endogenous antigen. It's digested, okay, and the peptide fragments using whatever enzyme method. All right, at this time of this occurring, MHC1 is being synthesized, okay, within the endoplasmic reticulum. Okay, now this is when the big change starts to occur. Your antigen peptides come into the endoplasmic reticulum where MHC1 is being made. They form a complex, so the antigen peptides will form to the MHC1. And at that point, that complex gets packaged into a vesicle, and the vesicle can work its way through exocytosis, inserting in the plasma membrane, and now it's ready to roll and go present itself to cytotoxic T cells. So that is how antigen presentation occurs, but sometimes the way immune system works is we need to bring some type of chemical messenger into play. Now, these chemical messengers are known as cytokines. So this is just a small protein or hormone that helps to regulate immune cell behavior. Okay, so it's not just it can increase it or it can decrease it. It can either stimulate or inhibit, like cell growth and differentiation of the immune cells, immune cell activity, and it's very heavily used. You've probably heard of maybe interleukin 6, interleukin 1, interleukin 7. They're all associated with being pro-inflammatory, anti-inflammatory. But they have some key features. One is they are secreted. Okay, so there's these free-floating proteins that are in our systems. All right, they're secreted by our lymphocytes, our APCs, some that are not even within our immune system, like fibroblasts, endothelial cells, monocytes. You have a version of these that are expressed in your skeletal muscle cells when you're exercising, and kidney cells. So these cytokines are not just exclusively saved for expression in immune cells. Now, they can act in autocrine and paracrine and occasionally in more of an endocrine way. So autocrine is what we discussed before, where it's on the cell that is secreted from them. All right, so they come back and interact on the same cell, whereas a paracrine is it's on a nearby cell within the vicinity. So we kind of talked about some of their functional roles, all right, and some of the big ones you can say they do is promote lymphocyte activation and proliferation. So this is more of like a pro-inflammatory response, all right. So if a cytokine that is telling the system, we need to ramp up this response to this type of infection, it can promote to the activation of the lymphocytes as well as their proliferation. So they're growing up on the population of cells. Now, it can also coordinate an innate and adaptive immunity. So this helps with basically deciding if the response is coming from that quick, rapid, innate immunity, or if it is coming from more of the adaptive immunity with expression of antibodies, activation of different types of cells from a response. This comes into play with our memory T cells that we discussed previously, as well as regulating inflammation. So I already kind of talked about if it's pro-inflammatory, but it can also be anti-inflammatory. So we don't want to have too much of an inflammation going on. And so these are there to help kind of regulate if it's going one way versus the other. And we ourselves, we have obviously medication that helps with this, and treatments where you get your autoimmune disorders, controlling of the immune system as well with transplants. So a lot of the chemicals we take can act kind of in a similar fashion that the cytokines would as well. And now another one that is not so commonly talked about but is also immune cell related is that it helps with stimulating blood cell formation in the blood marrow. Okay, so we kind of talked about how just crucial, blood flowing through our body, the cardiovascular system, as well as generation of B cells from the bone marrow is. So having these cytokines, they can either stimulate or kind of hinder the formation of these. Now, these guys are a communication network, so they're not really go off and they do whatever response they have to do. They don't do it a direct manner. It's more of an indirect. So it's kind of like the mailman coming to deliver a letter. They don't really do it themselves. Now here I just have a table of some examples. I won't go through all of them. Just here for you to understand that these cytokines, one can interact in a pro-inflammatory, so supporting inflammation, anti-inflammatory, stopping or reducing inflammation, and it can interact on a variety of different cells. Okay, so T cells, helper T cells, cytotoxic T cells, and it can also be produced by those same cells as well as produced by other macrophages and other types of cells like B cells, dendritic cells. So most commonly for a lot of screenings for pro-inflammation that you'll see is more of an interleukin-6. Interferons are also looked at as well. Seeing if interleukin-1 as well. So a lot of these are used kind of for research or clinical based evaluation of how our inflammation system is interacting. So they do play a crucial role in how it works. Now I want to get a little more into the exact method that the cell mediated immunity works. All right, so it's considered T cell responses. Now, cell-mediated immunity is driven by these T lymphocytes. Okay, and a lot of times it targets certain things. That's like virus-infected cells, our cancer cells, transplanted tissues, intracell pathogens, which we talked about a little bit before. So good and bad ways. All right, and so activation of the T cells has what is known as a two-signal role. All right, and most T cells are inactivated until they're properly activated, so they have to go through this signal role. Now, the first signal is antigen recognition, all right, which makes a lot of sense. We need to know exactly if there is a legit antigen and if it really needs a response, right? So T cell receptors, or TCRs, will bind to those antigen fragments that we covered about the presentation with the MHC molecule. So that cell that has processed with that antigen complex, MHC complex, has made its way into the lymphatic system, and then it finds these T cell receptors. All right, so the antigen was processed, presented, and now it's recognized. And so if you think way back when I was talking about different types of T cells, like helper T cells, cytotoxic T cells, they all had that own little recognition protein, CD4 or CD8, depending on the type. These guys act as co-receptors, which means they basically have an interaction with the receptor on another cell. So CD4 interacts with MHC2, CD8 interacts with MHC1. So that's your connection between which one is going to activate or impact which type of T cell. All right, so MHC2 to helper, MHC1 to cytotoxic. Now, the next step for the next signal is co-stimulation. Now, this is kind of like a check and balance. So it's required to prevent that there's any accidental immune activation. All right, so it's there to make sure that we're not just firing up an inflammatory immune response if there's no need to. This is when our cytokines start to come into play, kind of getting that conformational signal, adhesion molecules between the APCs and T cells, so kind of like similar co-receptor interactions. And if there is no co-stimulation, we have what's known as anergy. All right, so the T cells become permanently inactive until the two signal rule is refollowed again. All right, so the co-stimulation is kind of like that. If it goes through it, it will continue. If it doesn't, it will go back to starting position. All right, so this is where cytokines really come in to help with the full through with inflammatory response. Now, helper T cells, like I had said, are CD4 positive. Those are going to recognize your exogenous. And I said they're presented with MHC2 on those APCs, and they require the co-stimulation. Clonal selection for these guys, so if you remember, it's where they basically clone themselves with the specific receptor of the antigen. So they basically are ramping up the population that can increase the specificity of the antigen. These clonal selection undergoes with helper T cells, but it helps them differentiate into two different types of helper T cells. So your effector helper T cells and your memory helper T cells. And some of the key cytokines, especially one of the major cytokines that helper T cells, produce and kind of respond to is interleukin-2 or IL-2. So it's basically really essential for almost all immune responses. And when it is introduced, all right, and interacts with the helper T cells, it stimulates the T cell proliferation, the B cell activation, and the NK cell activity. So as you can see, it's acting as a pro-inflammatory cytokine, so it's increasing what is needed to allow an immune response to occur. And this acts via a positive feedback. All right, and so we can kind of see this when we're looking at HIV. All right, so HIV selectively destroys CD4 T cells. So in a response, we have kind of like an immune collapse, and it impacts the interactions of interleukin-2, which therefore does not really have that stimulation of T cell proliferation, B cell activation, NK cell activity. So it's kind of like the immune system has collapsed in on itself. Now, this is just showing you what I mean when I'm talking about that. We've already at this point have gone through the whole complex forming of MHC with whatever antigen, but this is where it comes into contact in the lymphatic system. All right, APC has this antigen MHC complex that is on its surface. It sees an inactive helper T cell that has a similar binding portion that can bind to the same antigen, but it also has that CD4 protein. All right, so this CD4 protein interacting with MHC2, that is your co-receptor. Okay, so they interact with one another. All right, so once this occurs, CD4 interacts with MHC2, the antigen interacts with both cells. This becomes an activated helper T cell. All right, so now that is when the clonal selection needs to occur. And so what happens is it decides, dependent on the antigen, if it's going to be expressing a certain receptor, if it's going to be secreting interleukin 2 in this example, or other cytokines, or if it's going to be more of a memory helper T cell where it's more long-lived, kind of dormant until there's a response. Okay, and for this instance, it falls on through to an active helper T cell, and that's when it will start secreting interleukin 2 or other cytokines, whereas memory, as we've seen, will mostly go for more of an adaptive response. Cytotoxic T cells, okay? These guys, as we've seen, are all about endogenous antigens, and this time with MHC1 instead of MHC2. And they actually require the interleukin 2 that comes from helper T cells for their full activation. All right, so without interleukin 2, we cannot activate cytotoxic T cells to do their job. And the colonial selection, similar to what we saw, it's going to result in two different types. All right, active cytotoxic T cells and memory cytotoxic T cells, which I'm assuming you can already guess what's going to happen with which. And this is very similar to, what we just saw. There's only like a minor change, okay? And that is, we've gone through it all. MHC1 has got its antigen. It's found into the lymphatic system. And what occurs is that infected body cell, because remember we took an endogenous epitope and we brought it to the surface, it allows the binding to an inactive cytotoxic T cell. Okay, so we have some interactions with the antigen and both receptors, the CD8 acting almost like CD4, has this co-receptor interaction with the other. However, once this occurs, it's still inactive. It's not considered active at this time. The activation comes with co-stimulation when the helper T cell, which has been activated, say similar to what our prior example was, it released interleukin 2. Interleukin 2 then comes in, and finally it becomes activated once the change in activation occurs. Now after it's activated, that's when colonial selection is allowed to occur. As said, depending on the response, it either goes to active or memory. So active will go forth and attack the affected cells, leading to its breakdown and the killing of the cells. Memory, again, it will stay with more so of like an adaptive response. Now, I've been saying that cytotoxic T cells are just the badasses that lead to the killing of the bad cells, the infected, the next target, but they have different mechanisms that they can use. All right, and they use all of them together, some of them together. It all really depends kind of if it's the memory cytotoxic T cell and it remembers the best way to fight this, or if it's just throwing it all out there to figure out which is the best way. Now, but the major killing mechanisms, one is granzymes. So granzymes go in basically tricking the cell into triggering the pathway that causes apoptosis or cell death. So basically tricks the cell into killing itself. The other is perforin. Now this one forms membrane pores. So this leads to cytolysis, leaky cells, or ruptures of cells. Okay, and granulosin is another one that destroys any microbes that are inside the target cell. So any type of cell that has a parasitic type interaction or virus. Lymphotoxin activates DNA destroying enzymes, so it's similar kind of to apoptosis, where it's almost instigating its own destruction. This time it's instigating its ability of the infected cell to destroy its own DNA. Okay, gamma interferon, this recruits and activates macrophages. Okay, so it's going to basically send out kind of like a chemotoxins cytokine. All right, leading to a recruit and activating of the macrophages. Now, it's important to note, though, these guys are all antigen specific, right? Because that's the whole point of MHC being this complex of the specific antigen and targeting cells and all that fun jazz. So it will not do its job unless it has that antigen and everything else that we discuss. The natural killer cells, they do not need to be antigen specific, and they are not. All right, so they're more of the wild card. So this is just a couple examples of exactly what I'm talking about when I'm saying the cell's kind of going forth and infecting it. If you have an activated cytotoxic T cell, it is bound to that infected cell, correct? Because it's all endo. Releases the granzymes. It gets into the infected body cell. It causes apoptosis, but like I said, all these immune cells assist one another, they feed off one another. The microbes are still going to be ruptured. So a lot of times what will occur is this phagocytes will be associated with such activated T cells. So they're there basically to anything that leaks out is what they're going to break down. Okay. Whereas in another instance, what can occur is there could be several being released, right? So you have granulosin and peripherin. And what occurs in that infected cells is it's going to destroy it by making like a leaky cell, all right? Cytolysis, where it's going to, pop. And this time we don't need a phagocyte because the granulosin is there to actually break down and destroy the microbes. So it can happen in a way without direct help from macrophages or phagocytes, or it can happen with phagocytes there to assist with the destruction of whatever microbe or infection. Okay, so as you can imagine, our immune system is just constantly just monitoring everything, right? It's like the security guards at the museum just making sure nothing's out of place. So it's just undergoing what is known as immunological surveillance. Okay, and it's really key. For instance, several cells a day will turn into a tumor cell or a cancer cell. This leads to a shift in expression of proteins either on the surface or secreted by some of these cells. So this immunological surveillance allows things like cytotoxic T cells, the NK cells, the macrophages to detect such shift in cells and eliminates them. Okay, so it has its ability to be very helpful. All right, this is why immune suppressed patients, they have a higher cancer risk because they are immune suppressed, like transplant recipients. Their immune system is reduced, so as a side effect, their body doesn't have this constant immunological surveillance in such an efficient manner to catch it before it's too late. Okay, so that was all cell mediated, a whole bunch of stuff, right? All right, so now we're going to talk about antibody mediated immunity. Okay, now where everything beforehand you saw was basically all T cells, a little bit of extra help from cytokines, things like that. This is driven by B cells, and these B cells remain in lymphoid tissue. Just like T cells, they have their own activation. It goes with antigens binding to a B cell receptor, antigen being processed and presented, this time with MHC2, and helper T cells providing co-stimulation to these cells via cytokines. Okay, so cytokines are coming back into play here. So helper T cells. Remember, helper T cells are the bridge across a lot of it. Now, just like the T cells, it goes through clonal selection. And it produces two different types. I'm sure you would expect memory is one of them. Now, they have effector B cells, but they're not called that. What they're called is plasmacytes or plasma cells. These guys are effector in the sense that they are there to secrete and produce the proper antibody that is needed to fight whatever infection is known by the antigen that's been presented. So looking at just a quick little, introduction of what we're talking about is, again, B cells are inactive, right? They have their receptors. Now, these receptors can recognize unprocessed antigens. Okay, so for instance, a microbe with specific proteins on its surface. And in the process of this occurring, it activates several B cells. Okay, so the B cells have become activated. However, we need further to kind of get the response to go. So what happens is helper T cells come into play. All right, B cells display a processed antigen that has come from collection of the microbes or breakdown, and it's recognized by the helper T cell. And so the helper T cell then, once it's recognized this antigen, it releases co-stimulators, aka cytokines. Now, once this occurs, the co-stimulation is there. We got clonal selection. All right, so the proliferation and differentiation of this activated B cell to decide, okay, am I going to make a memory for long-term dormant, or is it going to become a plasmacyte and do its job right away where it starts producing and secreting antibodies? Now, antibodies, I'm sure you're all familiar with, and definitely all heard the term. Antibodies are glycoproteins, okay, and they bind to specific antigens, as we've been talking about. They have a specific structure. There's variations, okay, in some of the structures as well as a couple regions of the structure. They have 2 heavy chains, 2 light chains, and I'll show a picture of what I mean by all this in a bit. And they have a variable region. Now, the variable region is given that name because it varies. It changes. So since it varies and changes, it allows it to recognize different antigens. So that is where the antigen is going to interact with the antibody. That's where it will bind, all dependent on what type of protein structure is there. And it also has what is known as a constant region. And the constant region helps with determining what type of class of antibody there is, because there's not just one size fit all of an antibody, there's a variety of different classes. So this constant region allows us to determine which class of antibody such response is coming from. And how antibody actions, I mean, as I'm sure a lot of you are familiar with, they protect the body in a variety of ways. All right, so they neutralize toxins and viruses, a lot of stuff they'll bind to. It'll prevent them from doing whatever type of issues within our body, and allows us to either pass them out or to be broken down later in the kidney. It can actually lead to immobilizing bacteria for movement. It can activate the complement pathway, which we'll get there, and it can enhance phagocytosis, so increase the ability of cells to, do the phagocytosis process. Now, this is what I mean when I'm talking about antibody structure. So this is probably the most common. You've probably seen it as a Y shape, okay? And they have, like I said, light chains. All right, so these are, if you're looking at the top of the Y, the inner or the outer sides of the 2 arms, okay? And then it has what are known as the heavy chains, okay? So heavy chains are here, okay? And then the antigen binding sites. So the antigen binding sites is basically in the middle of the Y. This is where it's variable, right? Because then it's going to determine which antigen it's going to end up binding to. And lastly, the stem region, which is also known as the constant region. Okay, so it helps us to determine which is which. And now, pardon my exclamation points for some reason, but each class has certain specific rules that it does. IgG is probably one of the most common, so that is there for long-term immunity. All right, so our vaccine response, our exposure to certain viruses like the flu and having an immunity towards it. It's huge in crossing the placenta. We talked about that a little bit, how it's more of like a passive immunity from mom to baby. IgA is a mucosal defense. All right, so it's there to help with some type of mucus response, such as saliva, vaginal secretions, as well as breast milk. Okay, so this is one that's allowed to cross from the breast milk into the baby. IgM, which is actually the first antibody that's produced. IgD, which is a B cell receptor. And then IgE. So IgE is one that's commonly associated with allergies and allergic reaction, as well as its response to parasitic infections. Okay, so each class has its own primary role, and a lot of times it's dependent on its structure because each structure of these are different. Okay, so as you can see here, IgG is the stereotypical Y, as we had just seen. IgA, it's basically 2 Y's stuck on one another. All right, tail to tail. IgM is just basically a flower of these Y's, okay? And IgD is basically an IgM with just slight variable changes, and there's not really a lot of it. It's mostly just on B cells. It's one of the 2 least. The least is actually IgE. So that's definitely not a lot there. Like I said, the most prominent is your IgG, followed by IgA and IgM. Okay, but they all have different locations, different functions. IgG is mostly in your blood, your plasma, and your intestines. IgA is also in your blood, but also, like I said, it's in sweat, your mucus, saliva, digestive secretions. IgM is back in blood, okay, in the lymph plasma, and whereas IgD, like I said, B cell receptor, and IgE is mostly on mast cells and basophils. So they all have their own function, and it is dependent on their structure, okay, and their location, obviously. All right, so throughout this entire thing, I've mentioned complement system several times, and now I finally will get to the complement system. So the complement system is just a cascade of plasma proteins that leads to an amplification of immune responses, and it can be an amplification in cell proliferation, cytokine production, antibody production, but all in all, it leads to an amplification of immune responses. And it does have some key outcomes that all dependent on the protein that is used. Okay, which protein ends? One is opsonization, so C3b. One is cytolysis via membrane attack complex, and the other is an inflammation. And now all of these pathways are dependent on activation pathways. Right, so we have a classical pathway that's most heavily studied. That is an antibody-dependent pathway that leads to this system to become activated. Then we have an antibody-independent one that is an alternative activation pathway. And then lastly, we have lectin. This pathway is more of a response to a carbohydrate recognition. It has a strong influence on our foods we eat and our intestinal lining, but also can lead to an activation of the same pathway. Now, the complement pathway starts with the C3 protein, okay, and the C3 is inactive, but it splits into two different forms. All right, it's activated C3A and C3B. C3b goes off, it binds to a surface of a microbe, okay, and receptors on phagocytes, so it attaches to the C3b. C3b goes and it enhances phagocytosis by coating a microbe, a process that is called opsonization. All right, so that's the one function. Okay, so it's gone through, split to become active. C3b goes forward, covers whatever microbe is there, and it helps to enhance phagocytosis by allowing the receptor of phagocytes to see C3b more. All right, so opsonization, first one. Now C3b also initiates a series of reactions. This brings about cytolysis. So C3b goes, there's another protein, C5, it splits to that guy. C5b fragment, then binds to two other proteins, C6 and C7, and that then attaches to the plasma membrane of a microbe. Right, C8 and C9 molecules join other complement proteins, and together they kind of form this cylinder-shaped membrane complex, which is called an attack complex, and it basically inserts itself into a plasma membrane, which allows creation of channels in the plasma membrane. That's what results in your cytolysis, or the bursting of the cells. Now lastly, C3a and C5a bind to mast cells, so another immune cell, and they cause them to release histamine. Okay, so we've already talked before about histamine being a key role when it comes to vasodilation impacting blood vessels. So histamine being released actually releases blood vessel permeability during inflammation. And C5a also attracts phagocytes to the inflammation site. So we've basically done vasodilation, and then we followed it up with our chemotaxis. Okay, so we basically have done the entire process of vasodilation and accessibility of phagocytes. Now this, again, just an example. Okay, we got an inactivated C3. C3 splits. It goes to C3b or C3a. C3b goes and it coats that entire microbe with itself, which can be detected by phagocytes, so that increases the phagocytosis, called opsonization. And then we have C5. Okay, C5 gets split again, C5b and C5a. C5a goes forth, helps to release histamine within mast cells. That then goes and increases vasodilation and chemotaxis for phagocyte cells. Now C6, C7, C8, and C9 come together to form basically just a giant pore, okay, that embeds itself into the membrane of the microbe, causing it to have a leaky membrane and as forth have the cell pop, basically. Okay, so I would kind of get to your brain, which really causes which, and which is the main one here. So just remember C3b, phagocytosis, C3a and C5a, histamine, vasodilation, 6, 7, 8, and 9, cytolysis or the attack channel. Now, we've been talking a lot about memory and vaccines and stuff like that. And so that all refers to what is known as immunological memory, okay, and exactly how it sounds. It's just the ability of your immune cells to remember an antigen it's seen before. So memory cells, which we've seen, we make memory T cells, we made memory B cells. They are there to allow a faster response, not just a faster one, but a stronger one, and to make antibodies they know that will have a higher affinity than what if it was this first response. Okay, and so when it's going through the immunological memory, it has what are known as a primary versus secondary response. All right, so basically it's first and second. So your first exposure at the time, it's slow, it's IgM, then it starts to release IgM, okay, or IgG. So it's the slow response of first exposure. All right, and then by the time you're exposed to it a second time, okay, so that's all the first exposure, you get exposed to it again later down, all right, it's more rapid, and it's mostly IgG because notice IgG was a successful response. So this is the foundation of vaccines. You get a light or dead dosage of a virus, all right, so what occurs is your primary response. And then what happens is if you get exposed to the virus, say the flu virus or COVID virus, then you will have more of a secondary response where it's more rapid and it's mostly IgG and correct antibody. So with the side of memory, we also need to be able to basically see this is me and also have our ability to tolerate whatever immune response is being thrown at us, right? Because we can have tissue damage in one portion of our body, but you're still getting the full wrath of inflammation or if there's an infection within one portion of the body. So what happens is we need to train the cells that are involved. And so most commonly it's T cells. Okay, this is called T cell education and it occurs in the thymus. And basically what it does is it teaches it a positive versus negative selection. So positive selection is T cells recognize their self MHC. Okay, so they recognize, okay, I have my MHC, so I am in fact a T cell that is active. Okay, so positive selection. Whereas the negative selection is on the flip side where it eliminates any self-reactive cells. Okay, so basically it needs to be able to control and recognize that it is an activated cell, and it also needs to know when it doesn't need to be activated anymore. Okay, so that's your negative selection port. Now, if this doesn't work, that's where we get our autoimmune diseases. So that cells can't recognize, hey, I no longer need to be on. We have stopped this response. We need to stop and, destroy the cells that are active. And there's clinical sides with this. This is like rheumatoid arthritis, type 1 diabetes. It comes with an issue with the immune cell's ability to say, I'm turned on, I'm turned off. All right, so like I said, the first thing we need to know is positive negative selection in the thymus. All right, so first question is immature T cell does it recognize itself MHC? If it says no, then the death of the cells occur. They can't recognize it, okay, because they need to know if they're on or off. Now, positive selection, yes. Okay, that's good. It can recognize itself. We're going to move on. Now, the next question is the receptor that's on those cells, is it capable of binding and recognizing self-peptides? If it says yes, deletes the death of T cell. If it says yes, it's inactivation of T cells. But if it says no, this is your negative selection, where the survival of the T cells that can recognize their self-MHC proteins is not determined on self-peptides as well. Okay, so they're able to basically know that they're on, but know that this is them. They can't attack them. Okay. Now, then if we have the mature T cells and lymphatic tissue, we have to be able to select the cells after they emerge. Okay, and this one's pretty easy. Okay, if there's no signaling occurring, then it's just death of the T cell. Okay, if there's no co-stimulation, but there's antigen recognition, then it just becomes inactivated. But if they detect that the cell can then recognize antigen and co-stimulate, the activation of T cell then allows proliferation and differentiation to occur. Now, B cells is very similar. Okay, it does immature B cell in the bone marrow recognize its MHC, just like T cells. If it does, then it's death. Okay, if it doesn't this time, so negative selection occurs first, actually. The mature B cell recognizes the antigen's first signal. Okay, so we have actually done the negative selection first. Okay, so once the mature B cell recognizes its antigen, if it doesn't have co-stimulation, it's inactivated. If it undergoes its co-stimulation, then B cell can be activated and it can proliferate and differentiate and do its plasmacytes function. So the B cell function of this, as you can see, is only a negative selection, whereas T cell side of things is a positive and a negative selection. Okay, now I just want to finish off again on just how the system impacts on other body systems. Okay, it's immune system, so I feel like you're going to see a theme. All right, now the first one is just our skin, our integumentary system. It allows for plasma proteins from dermis of skin to be leaked into interstitial fluid. There's specific immune systems or immune cells that are in the skin to help protect our skin. Obviously, it's some that are much tougher, like some versions of macrophages. And our lymphatic tissue applies or allows to IgA antibodies to be expressed in sweat. Now our skeletal system, again, the vessels are helping to drain excess interstitial fluid and leaked blood plasma proteins from connective tissues around bones. All right, so again, you'll start to see the vessels are one of the main kind of driver in a lot of these things. And the same thing happens in the muscular system. All right, so it's helping to drain any excess fluid and proteins from the muscles. Nervous system. Obviously, the immune system is super important here, right? We don't want our nervous system going down. So the immune cells really help to protect the nervous system from pathogens, okay? And the brain actually helps regulate the immune responses. So it's kind of like a feedback, a little interaction with one another. And just as anything else that we've seen, the lymphatic vessels help with the draining of the excess fluid and any type of leak proteins from nervous system. Neuropeptides that are associated with the lymphoid system, okay, and its response can also actually function as some neurotransmitter. So there's some tie over with the lymphoid and the nervous system interaction and firing. Now, the endocrine system, not only does the flow of the lymph plasma help to stimulate cytokines, but as we've seen in the previous slides how hormones were so important in that system, this flow of the lymph plasma actually helps some of the hormones also to be distributed throughout the body, as well as the vessels allowing for any type of leak of excess interstitial fluid from the endocrine glands. Now, we already know cardiovascular system is extremely important, okay, just like, it is filtering the fluid that is returning into the lymph from blood capillaries, all right, so it's helping to return blood as well. The macrophages and spleen destroy aged red blood cells and remove debris in blood. So it's this system is almost acting like a type of filtration and like TSA security type guards. Respiratory system. Okay, so our tonsils, macrophages in the lungs, the malt, those are all there to help protect the lungs from pathogens. We are breathing in some probably nasty crap at our time right now, right? Stuff that it can get in, it can affect our respiratory tract. If working in the coal mines, it can give you black lung or cancers, things like that. So these guys and these types of cells are there to kind of help it. Okay, and as anything, helps with draining fluid as well. Digestive system. So the tonsils and malt come back to play a role as well in the digestive system. They actually help defend any toxins or pathogens that penetrate from the digestive canal. Okay, so if you're eating some bacterial-ridden food, salmonella, some food poisoning, they are there to help defend those pathogens, get them out of you. The digestive system also provides IgA antibodies in our saliva and gastrointestinal secretion. So your saliva right now is floating with IgA antibodies. Okay, digestive system actually starts in your mouth. All right, so it's there to start the protection methods. Again, lymphatic vessels, they will help to drain excess interstitial fluid or anything from the organs of the digestive system, but they also help to pick up any absorbed dietary lipids and fat-soluble vitamins. Okay, this is important because it's kind of hijacking and allowing to go straight from the small intestine and transport them straight into the blood. Urinary system, lymphatic vessels, again, they drain any interstitial fluid from organs of the urinary system, but malt helps to defend against toxins and pathogens that penetrate the body via the urethra. Okay, so they're there to try and help against many types of STDs, any type of bacterial infection. So malt is one of the main defenders when it comes, because again, it is a secondary lymphatic organ. So normally it will be positioned in areas where pathogen exposure is more common. And lastly, the reproductive systems. Now, lymphatic vessels, again, draining the interstitial fluid from organs of the genitalia systems. You'll start to see if there's ever a question that has that, you should just say draining of fluids from organs of that system. Malt also, once again, helps. Okay, and this time it's not just the urethra, it's from the vagina and the penis with what other toxins and pathogens that may be penetrated. Okay, and females, sperm actually deposited in the vagina are not attacked as foreign invaders, and it's due to inhibition of immune responses with the help of the fact that the malt is right there. Okay, so the malt has its protection as well as kind of its assistance when it comes to fertilization. IgG antibodies can also cross the placenta to provide protection, developing fetus that we have talked about, and that all again comes from this flow, the lymph throughout, carrying the IgG antibodies and allowing the cardiovascular arteries and all that fun stuff that's connected with the placenta to fly into the developing fetus. All right, and lymphoid tissue provides the IgA antibodies to the milk of the nursing mother. Okay, so again, similar to giving women vaccines when they're pregnant, helps the IgG get into the fetus. Same thing as a thought when it comes to nursing. Okay, so there's studies done that have shown that babies that are not breastfed commonly have shown to be sicker more than babies that are breastfed. There's also association with if a mother eats a certain thing, say peanuts, there's a slight, not definite association that there's something going on that usually the child is less likely to have an allergy. Okay, and, studies are still going about that, but this, giving of the antibodies across several fields of development is quite important. Now, clinically, disease-wise, as we get older, our immune system starts to drip. As we undergo stress, our immune system starts to drip. Okay, it's probably why you get a little cold when it comes to getting close to final exams, or you feel like crap when it comes to final exams. Taking care of my kid right now, I'm stressed. I definitely have a cold right now. It all comes from stress side because our cortisol is suppressing immune responses. All right, so the cortisol increases from the stress, and it leads to a suppression in immune responses. Okay, and now when I'm saying stress, I'm not just saying pulling your hair out because you have to study for an exam. This also comes from if you're getting poor sleep, if you're not eating healthy. That's why, if you stay up till 3 in the morning trying to crash an exam, then you're going to feel like crap probably the next day, especially if you're not eating healthy. All right, and with aging, as I mentioned a long, long time ago, is our thymus starts to, involute on itself as we grow. It continues through our aging process. So what occurs is we get fewer functional T cells. All right, so we start to lose our vaccine's effectiveness. All right, again, that happens in general, which is why boosters are important. But that is a lot of why, a lot of time the natural cause of death is just flu, pneumonia. It's because the body has had basically not enough T cells for a response to actually have an effect on the body. Now, I just put some clinical disorders here, just for those of you that are more interested on the clinical side. We've already talked about HIV, and that's destroying of the CD4 T cells, which is impacting the immune's activation and response and all of that. Allergies are basically having an IgE antibody mediated hypersensitivity to a certain protein or fat that comes from whatever food or exposure to plants, anything like that, any type of response that leads to this hypersensitivity type immune response. Autoimmune diseases we talked about, it's just loss of self-tolerance. All right, so it can't tell itself, okay, you need to stop attacking, or hey, we need to attack. SID is when B and T cell function is basically absent or reduced. Lymphomas is the common cancers of all lymphoid tissues. Okay, and of course, some infections, mononucleosis, is just an EBV infection of B cells. All right, so some viral infections can occur in the immune cells, which impact its own function. Okay, and that so impacts the immune system. All right, so as I had said, all right, you have the quiz this week. Okay, the next week, no lecture is going to be uploaded. There will be an exam posted in the same spot the quizzes are. Same format, lockdown browser, 100 question, multiple choice. Okay, webcam, Wi-Fi. Give yourself at least an hour. Okay, there's no definitive time I have on it, but give yourself at least an hour and do it all in one shot. Okay, because if you leave, then it's just going to automatically submit it for you. All right, so do it all in one shot. So I will post the password when it gets closer to it, and I will make sure to send out an email to you all. All right, and feel free to email me or come to my office hours if you have any questions, want to go over some stuff. All right.